This video will compare and contrast hydrophilic and hydrophobic hormones. We will focus on how each class of hormone interacts with receptors and how target cells respond. Hormones can be broadly classified based on their solubility in water or lipids. This property determines whether a hormone binds to a cell surface receptor or an intracellular receptor. Here we see an important example of each. Epinephrine is a hydrophilic hormone, meaning it is water-soluble and binds to cell surface receptors. Cortisol is a hydrophobic hormone, meaning it is lipid-soluble and binds to intracellular receptors. Epinephrine and cortisol are both stress response hormones produced by the adrenal glands. While epinephrine is important for the rapid response to stress, cortisol is a long-term stress response hormone. In this video, we will use epinephrine and cortisol as examples to see how target cells respond to hydrophilic and hydrophobic hormones. Hydrophilic hormones are water-soluble hormones. Because they are water-soluble, they cannot readily cross the phospholipid bilayer of the cell membrane. As a result, hydrophilic hormones bind to receptors located on the cell surface. Most amino acid-based hormones are hydrophilic. Major examples include catecholamine hormones such as epinephrine, norepinephrine, and dopamine. Melatonin and serotonin are also modified amino acids that function as hydrophilic hormones. Peptide and protein hormones are also hydrophilic. This is the largest category of hormones. Major examples include parathyroid hormone, insulin, glucagon, the releasing hormones produced by the hypothalamus such as CRH and GHRH, and all hormones released from the pituitary gland. Growth hormone, prolactin, and FSH are examples of protein hormones released by the anterior pituitary gland. Oxytocin and ADH are peptide neurohormones produced by neurons in the hypothalamus and released from the posterior pituitary gland. Most of the cell surface receptors that are activated by hydrophilic hormones are G-protein-coupled receptors. When activated, the alpha subunit of the heterotrimeric G protein binds to GTP. The G protein dissociates from the receptor, and the alpha subunit separates from the beta and gamma subunits. These G proteins relay the signal into the cell by regulating enzyme activity. Different receptor types on the surface of target cells can activate distinct classes of G protein subunits, leading to a variety of cellular responses. The enzymes regulated by G protein-coupled receptors often produce or break down intracellular signaling molecules called second messengers. Inositol triphosphate, abbreviated IP3, is an important example of a second messenger. The enzyme phospholipase C produces IP3 from a phospholipid in the cell membrane that we can call PIP2. IP3 is produced when epinephrine and norepinephrine activate alpha-adrenergic receptors. IP3 binds to receptors in the membrane of the sarcoplasmic reticulum to release calcium-activating calmodulin and myosin light chain kinase, leading to smooth muscle contraction. This occurs when epinephrine binds to alpha-adrenergic receptors on the pupillary dilator muscles of the iris, and similar mechanisms regulate smooth muscle in other organs. Another important example of a second messenger is cyclic adenosine monophosphate, abbreviated cyclic AMP. The enzyme adenylyl cyclase converts ATP to cyclic AMP. Many hydrophilic hormones stimulate the production of cyclic AMP, leading to activation of protein kinase cascades as intracellular signals that regulate the physiology of target cells. Kinases are enzymes that add phosphate groups to their substrates. Protein kinases phosphorylate other proteins to regulate their activity. One of my teachers, named Donald Walsh, was the first person to discover a protein kinase intracellular signaling cascade. In this classic example, epinephrine stimulates the protein kinase A cascade leading to glycogenolysis. Glycogen is a polysaccharide composed of repeating glucose monomers and serves as an energy storage molecule in animal cells. Glycogenesis is the production of glycogen to store glucose in cells. Glycogenolysis is the hydrolysis of glycogen to release glucose. When epinephrine binds to beta-adrenergic receptors on the cell surface, the G protein activates the enzyme adenylyl cyclase. Adenylyl cyclase produces the second messenger cyclic AMP. Cyclic AMP activates protein kinase A, abbreviated PKA. PKA phosphorylates and activates glycogen phosphorylase kinase, abbreviated GPK. GPK then phosphorylates and activates glycogen phosphorylase, the enzyme that breaks down glycogen-releasing glucose in the process known as glycogenolysis. Glycogenolysis in the liver releases glucose to enter the blood. In contrast to hydrophilic hormones, hydrophobic hormones are lipid-soluble hormones. Most hydrophobic hormones are steroid hormones synthesized from cholesterol. Cortisol is an example of a steroid hormone important for the long-term stress response. Although most amino acid-based hormones are water-soluble, thyroid hormones are lipid-soluble. Here we see the structure of thyroxine, or T4, the thyroid hormone that contains 4 iodine atoms. All steroids are produced from cholesterol. This includes progesterone, testosterone, estrogen, cortisol, and aldosterone. While hydrophilic hormones are directly soluble in the blood plasma, hydrophobic hormones need transport proteins to carry them suspended in the blood. Because they are lipid-soluble, hydrophobic hormones can readily cross the phospholipid bilayer of the cell membrane and bind to intracellular receptors located in the cytoplasm or nucleus. A key example of hydrophobic hormone action is the stimulation of gluconeogenesis by cortisol. Gluconeogenesis is the metabolic pathway that produces glucose from non-carbohydrate precursors such as lactic acid. Cortisol can stimulate gluconeogenesis by increasing the activity of the enzyme phosphoenol pyruvate carboxykinase, abbreviated PEP-CK. This enzyme catalyzes the conversion of oxaloacetate to phosphenol pyruvate. This is the rate-limiting step of gluconeogenesis, so when the production of phosphenol pyruvate is increased, gluconeogenesis speeds up. Cortisol binds to glucocorticoid receptors inside the cell, and the complex enters the nucleus to stimulate transcription of the gene for PEP-CK. In response to cortisol, increased PEP-CK levels increase gluconeogenesis in the liver. This response provides a slower but longer-lasting increase in blood glucose compared to glycogenolysis. To recap, hydrophilic hormones are water-soluble molecules that act through cell surface receptors and second messengers. Their effects are typically rapid and short-lived. Hydrophobic hormones enter target cells and directly regulate gene expression. Their effects develop more slowly but tend to last longer. Together, these two hormone signaling strategies allow the body to respond to both immediate and long-term physiological demands.